in tumor cells, they start to express or hyperactivate this pathway, like EGF receptor or mutated RAS, activate MAP kinase, which phosphorylates a different region of the SMAD3. And now, because it's phosphorylating a different region, it will bind to different proteins and activate different genes downstream of that. And then, so the balance between the two, both are occurring at the same time. This is super low in normal cells. This is very high, so you get normal cells, apoptosis. This is incredibly high in tumor cells. This could be also high in normal cells, but because this linker region gets phosphorylated, you shift it to this side, i.e. the linker region phosphorylation, and activate different genes. So both are occurring. It's just a question of who's whispering and who's shouting within the cell. Okay. All right, and that's in normal cells, going to tumor cells. Now, TGF-beta, because you've altered the different genes that it's going to hit, TGF-beta does something else. It induces epithelial to mesenchymal transition. And you probably see something like this before. So basically, I'm just going to highlight some of the things that are important. We'll have normal epithelial cells. What is epithelial to mesenchymal transition? Well, it's when you start with an epithelial cell, and because you're affecting it with growth factors, cytokines, extracellular matrix, in this case, TGF-beta, they'll undergo dissolution of tight junctions. First, the gap junctions go, the tight junctions go. Then you'll have the desmosomes and the adherence junctions. They go as well. It's just a relative strength of these cell-to-cell communication junctions or adhesions. And as you lose your adherence junctions and your desmosomes, the cells become individualized. They alter their cytoskeleton, and they go from being nice, rigid epithelial cells to mesenchymal cells that can move over uneven terrain, can talk to cancer-associated fibroblasts, can chew their way through the extracellular matrix, break off from the main tumor, and then just go off and metastasize, right? And I think I've already mentioned, once they set up shop in a different organ after they've metastasized, you can do the opposite. You start off with your mesenchymal cell, and then there goes MET, mesenchymal to epithelial transition. And you start to have cell-to-cell contacts, and you usually stall at something like here. You never go back to becoming a normal epithelium. So if you have a skin tumor that detaches itself and goes into, I don't know, the lung, and makes metastases, it never becomes fully skin again. It doesn't differentiate into perfect skin. The origin is skin, it's a skin tumor, but it's still a blob of cells, growing, hyperplagic, hyperplastic, and dysplastic, and so on and so forth. So this perfect circle here is something like wound healing. If you scrape yourself, the cells have to undergo EMT because you have your keratinocytes, epidermis, and your epithelial cells in your skin. They have to detach from each other in order to migrate and fill the wound. And so they have to go to become mesenchymal. But once they fill the wound, they actually go back to becoming perfectly epithelial again. So wound healing is like controlled cancer, or cancer is like uncontrolled wound healing. Same processes occur. And so the way to, and you can actually follow along. Now the proteins that we talked about already is E-cadherin. I talked about E-cadherin in the EMT lecture. I talked about vimentin, which is a mesenchymal marker, in the EMT lecture. So you can actually follow along what's going on in these cells just by looking at them. And the cytokine that I talked about was TGF-beta. And I haven't talked about Rho GTPase activation just yet. I'll talk about it a little bit later. That's one way that you can sort of go back into epithelium. Let's look at EMT first. I have a feeling I already discussed this slide. Have I discussed this slide in the EMT lecture? No? Okay, good. So I'm going to go through it now. I've discussed it last year, I think, in 3140. That's why I got déjà vu. All right. So here, let's start off with murine mammary cells. On the top, and rat bladder carcinoma cells on the bottom. So what did they do here? They looked for a marker for epithelial cells, E-cadherin, we already discussed this, or you look for vimentin, that's a mesenchymal cell marker. And this is done by immunofluorescence microscopy. So you throw in DAPI. DAPI binds to DNA to tell you where your nuclei are. It allows you to actually focus in on your cells, because sometimes your signals are so weak you can't see them. But the second you have DAPI, you can easily focus in on your cells and then see if everything else is there. In this case, it's really nice expressed. So your E-cadherin is the cell junctions that go around the whole cell, make these belts around the cell. Keep in mind here, in this panel, they have both antibodies. They have the E-cadherin antibody that fluoresces in green. They have the vimentin antibody that fluoresces in red. The vimentin is just not expressed very well. That's why you can't really see it. Maybe you see a couple of dots here and there of vimentin. That's because there's a low-grade expression of vimentin. And that's an epithelial cell. But you add TGF-beta to these cells, and you force them to become mesenchymal. So what does that mean? Well, you lose your epithelial marker, so the ECAD is gone, or very little here, a little bit of green here, here, and here. A lot of red, which is your vimentin, which is your mesenchymal cell marker. So you know that you've actually transitioned from epithelium to mesenchymal phenotype, same cells. And the same thing can be true with other different epithelial cells. Here's desmoplakin. These are part of the desmosomes, another epithelial cell marker. Also makes these belts around the cells. You add TGF-beta, they reduce the expression of desmoplakin, increase the expression of vimentin, and now you have individual cells. And here's the question.
Sorry, earlier you said how TGF-beta, when you're adding it to normal cells, it's not causing like transformation.
Yeah.
In this experiment, when TGF-beta is added to the mammary cells,
yes,
we see that they're becoming more mesenchymal. So
yes, so these are tumor cells. Yeah, these are tumor cells. Now TGF-beta. Okay, so there's two things. One is TGF-beta will cause apoptosis in some cells, increase immune surveillance. They'll also induce EMT in a specific controlled environment, like in wound healing. So in wound healing, you don't have RAS mutations, you don't have RTK, you don't have linker region phosphorylation. But the cells will still respond to TGF-beta because other stuff is going on. So remember when I talked about PDGF versus EGF? Well, during wound healing, you don't just have TGF-beta there. You have TGF-beta, interleukins, TNF-alpha, and other cytokines. And other growth factors are released. So the combination of all those growth factors will actually induce TGF-beta to push the cells to undergo EMT, right? Even if they're like in a normal epithelium, they're not tumorigenic. So that's another nuance about TGF-beta. So TGF-beta can do three things. One, in normal cells, you want it to be intact to induce apoptosis. In tumor cells where you have receptor tyrosine kinases that sort of commandeer the TGF-beta pathway, and then you have this third potential EMT factor with TGF-beta, depending on who else is also, or what other cytokine is also there. I didn't want to talk about the third one in particular, and I like to lump some EMT with the tumor, because that's where you see TGF-beta effects. So keep in mind there are three effects that TGF-beta does in cells, but to keep it super clear in your minds for exam purposes and that sort of thing, I just split it into two. TGF-beta on its own, apoptosis. TGF-beta either because there's other growth factors or activation, hyperactivation of the pathway, EMT in tumor. Yeah. That's a great question, and it's a very nuanced. There should be a very nuanced answer, which does not allow you to get to a question on an exam like, I don't know what he's asking, because it could be this, it could be that. So I'd like to teach it in like black and white. Yeah. Keep in mind, there's gray zones everywhere. Yeah. All right, so remember when I talked about Rho GTPase activation? So that's how you can sort of take a mesenchymal cell and sort of force it back into an epithelial constraint or phenotype, and you play around with Rho GTPases. So one way that TGF-beta induces EMT is, I'll just draw it up over here because I don't have a laser pointer. So TGF-beta will bind to the receptor. Type 2 receptor phosphorylates type 1 receptor. We know that already. Activates SMAD. SMAD goes into the nucleus, binds some code factors, induces SNAIL. The SNAIL is a transcription factor that induces EMT. How does it do that? Well, SNAIL is a transcription factor that shuts down E-cadherin production. And if you have no E-cadherin, you don't have adherence junctions and desmosomes. Well, in this case, it's adherence junctions. And you induce EMT. But there's a second way to do it. And this is coming back to that SMRF protein. So earlier I mentioned how SMAD7 brings SMRF to the receptor. It's an E3 ubiquitin ligase. And because SMAD7 associates with the receptor with SMRF, SMRF ubiquitinates the receptor and degrades it. Right? But there's a second way to actually get SMRF to the receptor. And it's not through SMAD7, it's through a protein called PAR6, partition defective 6. So PAR6 will also bind to the type 1 receptor. And now what's interesting is that if SMRF is associated with the receptor complex through PAR6, not through SMAD7, SMRF changes its target. It doesn't go after the receptor anymore. It'll go after Rho GTPases and cause Rho, small GT binding protein, to be degraded. Let me draw that up. So, you have the receptor. Let's say it associates with SMAD7. SMAD7 and SMRF6. No, SMRF2. SMRF. Or could associate with PAR6 and SMRF2. In this context, SMAD7 binds the receptor, blocks it from accessing SMAD2. SMRF2 ubiquitinates the receptor. And in the receptor complex, degradation. In this context, SMRF2 does not ubiquitinate the receptor. It looks for Rho, which is in the membrane. So SMRF ubiquitinates Rho, and this gets degraded. Just Rho gets degraded. So once again, you have the proteins doing different jobs depending on who they're interacting with. So in this case, SMRF is the same SMRF protein, but the way it interacts with the receptor is important. If it interacts with the receptor through SMAD7, the receptor gets degraded. If it interacts with the receptor through PAR6, Rho gets degraded. Question?
So, like in what scenario would it be interacting with PAR6 instead of?
So it's a question of timing. So the receptor normally goes through the SMAD2 pathway. After a couple of hours, SMAD7 comes up with SMRF, causes the receptor degradation. Any receptor that's left over, after a couple of days, PAR6 comes up with SMRF and then activates or hits Rho. Yeah. Not all the receptors are targeted because there's only a finite amount of SMAD7 in the cell. Good question. So it's a question of timing. Signal first, then SMAD7, then SMERT2 with FAR6. And what happens if you get rid of Rho? You also get tight junction dissolution, and you block EMT, or you target EMT. So you come at it from two different angles. One is through tight junctions, and the other one is through adherence junctions. And snail, of course, also increases vimentin, which is a different cell cytoskeleton protein compared to keratin, for example, in the skin, or another rigid intermediate filament protein. All right. Now, what are TGF-beta receptor therapeutic targets? Well, you can go after it different ways. Here's a nice sort of summary of how you can do it. Starting from cells in culture, going to animal models. So these are the biomarkers in TGF-beta cancer drug discovery that you can use. So what you do is you start off with the purpose, biochemical activity of the target. So you can look for kinase inhibition. We don't have very good phosphorylation antibodies for the receptors. We've tried, different labs that I've been in tried, and the field itself has tried. And we can't find good antibodies that recognize as phosphorylated receptors. Really hard to do. So instead of going after a phosphorylated receptor to see if you've actually inhibited the receptor, what you can do is you look at SMAD, or phosphosmad2 and phosphosmad3. Let's do it this way. SMAD2, SMAD3, phosphorylation. You could make an antibody to phosphosmad2, phosphosmad3. So you look at phosphosmad analysis in peripheral blood mononuclear cells and tissues, for example, in tumor biopsy. I'll show you that in a second. You can look for signaling pathway analysis. SMAD2 and 3 will associate with the common SMAD, SMAD4. So you can look for co-immunoprecipitation assays, which I'll describe that in greater detail next week. You can look for gene expression. You can look for secreted proteins in response to TGF-beta signaling. So that's what you can do. These are the parameters, and this is purely biochemical in cells. And then you can start to look at, evaluate the cancer endpoint. Apoptosis, proliferation, angiogenesis, immunosuppression. By looking at different proteins by immunohistochemistry, EMT by immunohistochemistry or immunofluorescence microscopy, vimentin, and E-coherent, for example. And then eventually go into the disease process, tumor growth, functional imaging, metastasis survival. And then surrogate to clinical benefit, objective response. Do the animals fare well if you inject xenograft tumor? And these are available clinical endpoints. Before I move on, I just skipped over your question. I'm sorry.
Oh, no, it was just about the PAR6. I was also asking about that, like if it would be similar to SMAD7, like coming from the nucleus.
Yes.
Going up. Okay.
Yeah. Great. Sorry about that.
Okay.
Just all right, so how do you actually do this? Well, here's an example. Let's say you take a mouse, you implant the tumor, and then you administer a compound, orally, intravenously, subcutaneously. And the xenograft, these are the tumor cells that are expressing TGF-beta. And because this, you want to go in with the TGF-beta inhibitor, so you go in with the tumor that you know overexpresses TGF-beta. And you take out either the plasma. And the reason you take out the blood or the plasma is because you want to see how much of the compound is there, especially if you administer something orally. Did the liver break it down? Does it get absorbed? What is the bioavailability? What is the pharmacokinetics and dynamics within the body? So you can follow the concentration in the blood, and then you can also see what does it do to the tumor. Does it slow down the growth of the tumor? Does it stop the phosphorylation of SMADs? You actually block this pathway in the tumor. And here are examples. So here's the concentration of T-beta-1 inhibitor administered to the animal. You have low, intermediate, and high. Low is so low you can't even see it in the blood, shown over here. It's a tiny little blip here. Intermediate levels, you can actually find it concentrated in the plasma. And then the highest concentration should give you the highest amount. And it'll tell you also how long does it stay in the bloodstream for. So that's taking the plasma, doing concentration, but you can also do phosphorylation of SMAD2. So here's phospho-SMAD2. If you have the vehicle control in the tumor, here are 4 different tumors that has lots of phosphorylated SMAD2. And then if you have low levels of the inhibitor in the bloodstream, it's not getting to your tumor, so it can't block phosphorylation of SMAD2. Intermediate amount makes it to your tumor and actually starts to block phospho-SMAD2. And then the highest concentration does inhibit phospho-SMAD2. Of course, they're lacking SMAD2 blots, actin loading controls. There are a couple of things missing from this image here. But you get the idea. You increase the concentration of the inhibitor, you go from no effect on the tumor to reduced phosphorylation of SMAD2, indicating that you're actually inhibiting the type 1 receptor. And what are the different inhibitors that we've come up with over the years? Well, there's antibodies, just like the EGF receptor. We have antibodies, but in this case, the antibodies are not to the receptor, the antibodies are to the ligand, right here. Or you have kinase inhibitors. These are not tyrosine kinase inhibitors, these are serine threonine kinase inhibitors, right? Because these are serine threonine kinases. The kinase inhibitors still look like ATP, but they'll fit into these ATP binding pockets a little bit differently from the tyrosine kinase inhibitors. Or there's antisense oligonucleotides that actually target TGF-beta itself. Now what's interesting about TGF-beta, unlike EGF receptor family, which has EGF, HER-regulins, neuregulins, TGF-alpha, and they bind to different receptors, except for HER2, or because that has no known ligand. TGF-beta is a little different. It has TGF-beta1, TGF-beta2, TGF-beta3 ligands. 3 ligands, beta-1 and beta-2 are so similar, the same antibody will kill both of them. And you can get antibodies that will also recognize TGF-beta3. So one antibody will target all the TGF-beta ligands. And therefore, you can also use this AP1209, which is an antisense oligonucleotide, to target the mRNA for TGF-beta1 and 2, because they're so similar. They come from the same gene product. So that makes life a little bit easier. So there's a lot of things here. I'm going to talk about a couple of these. So here are the strategies and where they are in phase 1, phase 2 clinical trials. Hilarious, 20 years ago is where this review came out. And I'll show you next week. There's another review that just came out a few months ago. They have the same idea, except they've moved from like phase 1 to phase 2, or discontinued, or finally we're recruiting for phase 1. So it takes a long time for these to actually make it to market. So this slide, although it's dated in terms of time, it's not dated in terms of functionality and the information that's on the slide. So the TGF-beta antisense oligonucleotides, we're still in phase 2 and phase 3 back then. Some of them actually are not, they discontinued them, or they pulled the actual clinical trial. Dominant negative receptors, that really never took off. Soluble domains, they're okay, as I'll show you next week. And small molecule drugs, they're still in phase 1, phase 2, even after 20 years, or 19 years. So let's talk about some of these inhibitors. SD208, it's a kinase inhibitor, it's the ALK5. Unfortunately, in every field, you'll have different people cloning the same protein and then coming up and saying, oh, T-beta-1, oh, that's the same as ALK5. So the type 1 receptor is T-beta-R1, or TGF-beta receptor 1, also known as ALK5, activin-like kinase 5. It's the same protein. So SD208 binds to the type 1 receptor, blocks the type 1 receptor. And if you look at the crystal structure of this inhibitor, it occupies the ATP binding pocket. So the same way that the EGF receptor inhibitors fit into the ATP binding pocket for the EGF receptor, the type 1 TGF-beta receptor inhibitor finds its way into the binding pocket of the type 1 receptor. Interestingly enough, the closest relative to the type 1 receptor is the type 2 receptor, and the same ATP inhibitor will not fit into the ATP binding pocket of type 2. It's really specific for the type 1 receptor. And this slide is just to remind you, to tell you that these are serine-threonine kinases, not tyrosine kinases. The job of a kinase, all kinases, is to have an ATP binding pocket. So you have to be mindful of this when you make ATP analog to inhibit kinase activity, to make sure that it's specific. And like I said, SD208 is really specific to the type 1 receptor. So here it is. So you have an ATP binding, it's an ATP competitive inhibitor, very small, makes sense. It has to look like ATP, it's quite small. Bicyclic ring, third ring over here, this sort of looks like ATP. Once again, don't memorize the structure, please. It binds to the transforming growth factor receptor 1, TGF-beta R1 or T-beta R1. Really small IC50, 49 nanomolar, which is great. And it shows 100-fold selectivity over T-beta R2. So it's 100-fold better at inhibiting type 1 receptor than its closest relative, type 2 receptor. And it has anti-inflammatory and anti-tumor activity. And here's an example of how it can be used. So SD208 inhibits SMAD3 phosphorylation and SMAD3 specific gene transcription. So let's follow this a little bit. So what did they do here? They took it's called lung, but they're not really lung. These are melanoma cell cultures. They're found from a lung node. So these are human melanoma cells. They were cultured in the presence or absence of SD208 and TGF-beta, 5 nanograms per mil for 30 minutes. So if you take these cells and you serum starve them, there's nothing in the serum that, like TGF-beta, for example, that will activate the pathways. There should be very little phosphorylation of SMAD3 if you use a phospho-SMAD3 specific antibody. Here's the SMAD3 and here's the beta-actin loading control. But you go in and you dump in 5 nanograms per mil of TGF-beta, and you have phosphorylation of SMAD3 shown in the second lane. So that's good. So minus and plus beta, you have nice phosphorylation of SMAD3. And you can start titrating in 0.1, 0.5, 1, and 5 micromolar SD208. And you can see a decrease in the phosphorylation of SMAD3. And at the highest concentration, 5 nanomolar, micromolar, excuse me, even at the 1 micromolar, it's very similar to the basal condition, where there's no TGF-beta added. So what they did here was they used a phospho-SMAD3 antibody that recognizes phosphorylated SMAD3. That's great. Now, if they're inhibiting phospho-SMAD3, SMAD3 cannot associate with SMAD4 and cannot go into the nucleus and induce transcription. Now, how can you follow gene transcription? Well, you can actually, instead of looking for an endogenous gene that's being transcribed, you can just transfect in a plasmid that has luciferase on it. With a specific promoter, a promoter of your choosing. And in this case, they chose a promoter that has a lot of CAGA sites. CAGA promoter. And the reason why you want CAGA is because those are SMAD3 binding sites. So if SMAD3 is phosphorylated and associates with SMAD4, it'll go bind to this promoter, SMAD3, SMAD4. It'll make this protein, luciferase, and it'll give off light. So this is CAGA, CAGA 9. So there's 9 times 9. There's 9 of these sites within the promoter in this vector. And you see that in the absence of TGF-beta, they don't produce any activity. There's no luciferase activity. There's no light given off when you put in a substrate. But you add TGF-beta and it goes up. And this is the absence of an inhibitor. This just goes to show that this artificial system in your cells, because you transfect this into your cells, is actually working properly. But now you add 0.1, 0.5, 1, and 5 micromolar of SD208. And as you block phospho-SMAD3, phospho-SMAD3 cannot associate with SMAD4, cannot associate and activate the transcription of luciferase, and the amount of light produced gets lower and lower and lower. So you prove that transcription is inhibited with this inhibitor. So the TGF-beta are one kinase inhibitor, SD208, antagonizes TGF-beta signaling and gene responses in vitro, right? In vitro. Now let's take a look in vivo. So how did they do this? Well, they took mice and they inoculated with these melanoma cells. And melanoma cells love to home in onto bone. Remember when I talked about the blood flow patterns versus the seed and soil hypothesis? For some reason, melanoma cells love to undergo the seed and soil hypothesis or seed and soil mechanism and home in onto bone. And if you take X-rays, you'll see that this is where the melanoma actually seeded itself and it chewed bone. So there's like sort of a gray zone there. That's where bone should be, but it's not there because the tumor is there chewing away the bone. So the mice were inoculated with these melanoma cells. 12 days later, osteolytic lesions were detected by X-ray, and the mice divided into 3 identical groups. So group 1, group 2, group 3. They received either vehicle, group 1, that's why you see the biggest tumors right there. Group 2 got 20 milligrams per kilogram daily of SD208. So the tumor should be a little bit smaller, but it doesn't really look that much smaller than the control. Or 60 milligrams per kilogram. And here, I think you can see that the tumor is actually smaller in this case. And so you inoculate the animals. After a week and a half, you add the treatment, either the vehicle, the 20 or the 60 milligrams per kilogram per day, and then you follow the growth of the tumors by the lesions inside the bone. The vehicle control and the lower concentration of SD208 actually had quite large lesions. The tumor was still growing, but the higher concentration had the smallest lesions. So SD208 inhibits bone metastasis of these melanoma cells. That's the conclusion they came up with. Any questions so far? I think it's pretty straightforward. Okay. So this is preclinical. Next steps, phase 1 clinical trials. This was done not too far back. SD208. It's a great inhibitor. Now, that's one way to go after. Go after the kinase activity. What about going after the ligand before it can even bind to the kinase? Does that work? So this is 1D11 or GC1008. I'll call it GC1008. And GC1008 is an antibody. It's a pan-TGF-beta antibody. It binds all 3 ligands, TGF-beta 1, 2, and 3. They're in phase 2 clinical trials. In 2011, I'll pull out the paper next week, or the review article. It says that they're like still in phase 2, phase 3 clinical trials for the same drug. And so this is a wall of text. So let's go through it slowly. So this is the phase 1 and 2 study for GC1008. human anti-TGF-beta monoclonal antibody, patients with advanced malignant melanoma and renal cell carcinoma. So these are patients that had advanced disease and failed at least one prior therapy, and therefore they're being treated with GC1008 at 6 different doses. They found that there were no dose-limiting toxicity occurred within 20 days of the first dose. So if no dose-limiting toxicities were found, they were continued on 3 additional doses. And the tumor response was determined by RECIST, which is a response evaluation criteria in solid tumors. This is sort of a standardized criteria to see if the tumor is actually getting smaller. Extended treatment was offered to patients achieving stable disease. So if they responded properly or appropriately to the drug with no side effects, then they were offered, do you want to continue on it? And then the patient said yes or no. And these are the results. 22 patients were treated in phase 1. So this is a very small cohort. And the point of this study is to see if the drug hurts the patients. That's it. It's not to see if the patients respond to it. It's just, does it make them sick or sicker than they already are? So no dose-limiting toxicities are found, even at the highest concentration. They figured out that 15 milligrams per kilogram was a safe dose. Adverse events reported at least possible related to GC1008, including skin rash. Once again, anything that is going through a cell cycle is going to be affected. 9 out of 22 patients reported serious adverse events, which is not great, most of which were related to underlying disease. Yeah. And so far, 5 patients received standard or better than received extended treatment. Of these responders, 3 to 5 patients had mixed responses. So the conclusion was that GC1008 is well tolerated in neutralizing TGF-beta. It holds promise, and phase 2 clinical trial was thought to be worth trying with metastatic disease. This was in 2008, and then in 2014 they finally published this study. So this study came out in 2008. The abstract presented at a conference. And it was only published in 2014. The abstract, or the study was published in 2014. Just to give you an idea of how long things take to go from, we invited patients to apply to be a cohort. They went through the exclusion process to find, or the inclusion process to find which patients to treat. They did the treatment. They followed them for a certain period of time. They amassed the data. They published the data, or presented the data at a conference. And then finally, it took another 6 years to publish it in the paper. Now, is this the best way to go about it? Or is this targeting the TGF-beta ligand the best way to go about targeting TGF-beta effects within a tumor? Yes and no. It depends, really. So normally, TGF-beta is produced by a number of different cells. You'll have paracrine expression, platelets make TGF-beta, immune cells make TGF-beta, but the tumor itself also makes TGF-beta. And what happens is if you don't have an inhibitor, all of these effects or all of these sources of TGF-beta will impact on the tumor. And then if you add a TGF-beta inhibitor, you're blocking, because it's an antibody, it'll sop up all the TGF-beta in circulation. It'll sop up the ones that are from paracrine effects, and hopefully it'll decrease motility and survival in EMT. It'll help with immune effectors, and sometimes the problem is the TGF-beta the tumor produces, you have to try to sop it up as soon as possible before it actually can go and bind to its own receptor on its own cell surface. And the best way to do that is not to go with an antibody. The best way to do that is try to target the mRNA that's making the TGF-beta within the tumor itself. And that's where AP12009 comes in, this drug right here. Now, it's an antisense oligonucleotide. It's specific for TGF-beta 2, which is the most highly expressed within tumors, but it'll also target TGF-beta 1 mRNA. And the best place to look for it is in gliomas. But it's not the only place, as shown in the next slide. I think what I'll do is I'll do this as the last slide today, and then I'll pick up next week. Because I don't know if you noticed, I called this lecture 4 and 5. That's because I gave you all the slides for today and next week after the quiz. Yeah. So, here we have pancreatic tumor cells. They're called PA2, pancreatic tumor, that were seeded into a well. They start off like a little ball. And then you wait 17 hours, 24 hours, or 65 hours. They sort of spread. They sit down in the well, and they start to spread and grow outwards. That's shown right here. Now, these are tumor cells that respond to TGF-beta. They make their TGF-beta, but they also respond to exogenous TGF-beta. So if you dump a little bit of TGF-beta on top of these cells, they start off at the same place, but after 17 hours, you see that this is already larger. After 24 hours, much larger than the control. And after 65 hours, they fill the whole screen here. The whole micrograph is full of cells. So indeed, they do respond to TGF-beta. Now what if you take these cells here on the left and incubate them with a monoclonal antibody to TGF-beta 2? Can you actually block tumor growth? So what you're comparing is the third column to the first column. You start off with the same size. After 17 hours, it looks more or less like the control. 24 hours, it looks exactly like the control. After 65 hours, it looks very similar to the control. So the antibody that they put into the wells is not actually blocking tumor growth. And the idea is that the tumor's producing TGF-beta so fast and secreting it, and it's binding to the receptor at the cell surface faster than the antibody can see it. So how do you get around this? Well, instead of going with an antibody to try to target the ligand, let's try to stop the ligand from being made in the first place. Let's add AP12009, which is an antisense oligonucleotide. It finds the mRNA for TGF-beta 2, binds to it, causes it to be chewed up by dicer, kind of like the whole siRNA, small microRNAs and that sort of thing in the cell. So you chew up the mRNA, you cannot make the ligand. So these cells are transfected with this antisense oligonucleotide at the start, 17 hours, 24 hours, very similar to the control, 65 hours, very similar to the control. If you block the production of TGF-beta in these cells, you block the growth and the expansion of these cells. But trying to block it by going with an antibody to TGF-beta 2 won't do it. The cells produce the ligand and binds to the receptor faster than the antibody can see it. And this is a great place to stop for today. And next week I'll pick up how you can use this in patients.